Hello guys, so today we shall be discussing another important part in the virology, that is HH3 virus, that is also called as human herpes 3 virus or the Varicella Zoster virus, right? So we shall be discussing today human herpes 3 or Varicella Zoster virus, Varicella Zoster virus, VZV, we simply call it as VZV. So what is the important thing about this human herpes 3 or Varicella Zoster virus? So this virus is basically known to cause two different diseases, right? So what are those diseases? Disease number 1 and disease number 2. So disease number 1 over here is, this is known to cause chickenpox. This is known to cause chickenpox, and on the other hand it is also known to cause herpes zoster. It is also known to cause herpes zoster. So these are the two different diseases which this virus basically causes. So let us discuss how these diseases are caused, right? Let us discuss about the pathogenesis and about the virus in much detail, okay? So first of all, if you look at the structure of this virus, so what is the basic structure this virus consists of? So this is a double stranded DNA virus. So this is a DNA, but what DNA? It is a double stranded DNA virus. I mean, this is the viral DNA over here, okay? Surrounding that, you have got a layer which is double layered. You have got a layer like this which is double layer, right? When I zoom in, I will find it as a double layer, but in fact I will look it as only a single layer. So this double layered thing which you can see surrounding this viral double stranded DNA, this is called as your capsid. Now surrounding the capsid, you have got another important layer like this, okay? So this layer surrounding the capsid, where it has got some glycoproteins over here, right? So in this way. Let me draw a couple of them. Yeah, these are the things. So you have got some glycoproteins and all of these things like this. This is called as the lipid membrane. So let me mention all of that. This is your double stranded DNA, that is your viral DNA. That is your viral DNA. Okay. So this green color one is called as your capsid. And finally, this is called as your lipid membrane. Lipid membrane. This is called as your lipid membrane. Now, basically, how does this virus enter into your body? What is the transmission route? So, if we discuss about the transmission of this virus, right? If we discuss about the transmission route of this virus, basically this virus transmits through the respiratory droplets. For example, if someone coughs or sneezes, right? So the virus through these respiratory droplets enter into the person standing next to him, right? So through the respiratory droplets, basically this virus transmits through respiratory droplets, okay? Not only respiratory droplets, even by kissing, and not only by kissing. For example, if someone coughs or sneezes, this virus enters or lands up near the open cut surfaces of your skin as well as mucous membranes. So direct contact with this virus, direct contact through an open cut surface of your skin or mucous membranes. So open cut surfaces, right? Open cut surface, or I mean the direct contact, or by direct contact. Next important thing is that when someone coughs or sneezes, if the respiratory droplets fall into your eyes, even through the conjunctiva also they get transmitted. So even through conjunctiva also they get transmitted, right? So let us say right now this virus is in your upper respiratory tract, okay? Now how it is gonna infect, how it is gonna spread from one person to other person, whether this virus once if it enters into my body, before I sneeze out, it is going to come in a single number, or it is going to replicate in my body and then going to come out. So all of these things we shall be discussing right now. So let me draw a picture. First of all. Let me draw the lateral surface of the face and explain you how this virus is basically transmitted. Right. So all of you know this is your nasopharynx and this is your oropharynx. Nasopharynx and oropharynx together they what? Together they join together to form laryngopharynx, right? So let us say, for example, I cannot exactly show you here, but roughly let us say we have got you have got your tonsils over here. So these are the tonsils, just for an example, okay? These are the tonsils. I mean, anatomically it might be wrong. These are the tonsils here, okay? So this will be your nasopharynx and this is your oropharynx. Now lining within your nasopharynx, oropharynx or the laryngopharynx, whatever it is, lining within that you have got some epithelium over here, right? These are called as respiratory epithelium. And what type of respiratory epithelium is this? The type of respiratory epithelium is ciliated columnar epithelium. So this is ciliated columnar epithelium. So what I am going to do is that I am going to expose one ciliated columnar epithelium. I am going to draw it big and I am going to explain you how things gonna happen here. So let me draw one ciliated columnar epithelium over here. So first of all, this is the wall. This is the wall of the pharynx and this is your ciliated columnar epithelium, right? So this wavy pattern which I am drawing, this is called as the apical membrane. This is the apical membrane. So right now, where is the virus located, guys? Where is the virus right now? The virus is in the lumen here. This red dot represents the virus, okay? So what I am trying to explain you is the virus is here. So this is the double stranded DNA virus, right? Surrounding that you have got the capsid over here and surrounding the capsid you have got what? The lipid membrane over here. Now what happens is that this virus has to enter inside the cell, inside your ciliated columnar epithelial cell. Now how will it enter inside? How will it enter inside is that first of all the lipid membrane, the outermost layer of the virus is the lipid membrane. First of all the lipid membrane fuses with the apical membrane, and when it fuses with the apical membrane, how the picture is gonna look? Look here, I am going to draw it in the next slide over here. Look here, I am going to draw it in the next slide over here. Yeah. This is how it's gonna fuse. So let us say the lipid membrane has fused with the apical membrane, and what did it deliver inside? There is DNA over here now. This is the viral DNA, right? Surrounding that you have got the capsid. Did you understand now? What has happened is that the lipid membrane fused with the apical membrane. So once it fused with the apical membrane, the remaining contents are the viral DNA and the capsid. So the capsid and the viral DNA are sent inside the cell. Now what will happen? What will happen is that inside the cell, you have got nucleus, right? Let us say that this is the nucleus inside the cell which you have got. Now this black color thing which I have drawn just now is the nuclear membrane, okay? Now inside the cell, you have within the nucleus you have got the DNA. So this is the DNA of your own body. This is the DNA of the ciliated columnar epithelial cells, I mean, which is present in your own body. This is your body DNA. Now what will happen is that this capsid along with the viral DNA goes and fuses with this nuclear membrane. Now how it is gonna fuse with this nuclear membrane? Look, it goes all the way like this and it starts fusing with the nuclear membrane. When it starts fusing with the nuclear membrane, how does the picture look? So the capsid has fused with the nuclear membrane right now, and what it has done? it has left the DNA enter inside the nucleus. Okay. So, what is this, guys? No need to confuse. What is this? This is your nucleus. And what has happened? What has happened is that capsid fused with nuclear membrane. Capsid fused with the nuclear membrane. And right now, your viral DNA is inside. So, this one is your viral double-stranded DNA. Viral double-stranded DNA is inside. Now, what will happen is that this double-stranded viral DNA, right, this double-stranded viral DNA will fuse with the DNA of the host. Host in the sense, the individuals, the people, right, the patient who is infected with this, the person. So, this viral DNA fuses with the host DNA. So, when it will fuse with the host DNA, look what is going to happen. So, this is how the viral DNA is getting fused with the host DNA. Now, once it fuses with the host DNA, the viral DNA manipulates, right, manipulates completely the host DNA in such a way that this DNA right now, the main function of DNA is to code on mRNA, right? What is the main function of DNA? To code on mRNA. And what is the function of mRNA? To synthesize proteins. But in this case, as a viral DNA has completely manipulated your host DNA, so this mRNA is gonna release viral proteins, not the normal proteins which will be useful for your body, but those proteins which it is going to release right now are the viral proteins. So, it is going to release lots and lots of viral proteins. I mean lots and lots of capsid molecules in this way. It is going to synthesize lots and lots of capsid molecules. So, this virus is using our host DNA to synthesize its own proteins, right? So, these are the capsid molecules, and obviously, you know, the DNA also replicates. When it replicates, the viral DNA also duplicates. So, in the same way, you will have lots and lots of viral DNAs also in this way. So, now see, lots and lots of capsid proteins along with the viral DNAs have been synthesized. And one very important thing which you know, that we have also got another important thing, that is Golgi complex, right? So, within the cell we have got something called Golgi complex. Let us say this is your Golgi complex. This is your Golgi complex, let us say. Now, this capsid, newly formed capsid, along with the newly formed viral DNA, they fuse with the what? They fuse with the Golgi complex. Now, when they fuse with the Golgi complex, what is going to happen? What is going to happen is that, look here. So, this will be your viral DNA. Surrounding the viral DNA, you have got the capsid. Now, this capsid will move little bit forward. And again, this is your capsid and this is your viral DNA. It will move little bit forward, and this is your viral DNA, and surrounding this is your capsid. Now, the final end product is, what does it do is, within this Golgi complex, you have got the lipid membrane. So, this capsid or this entire virus gonna take that lipid membrane from the Golgi complex and surround itself. Look what is it going to do. So, this is your viral DNA. Surrounding this is your capsid, newly formed capsid. Now, this Golgi complex is having lipid. So, I already told you, surrounding this virus here, you have got the lipid layer. So, from where this lipid layer is going to come? From the Golgi complex. So, this virus will take a part of the lipid membrane from the Golgi complex and cover itself out like this. Okay. So, in this way, many viral DNAs are produced. I mean, many viruses are produced. In this way, many viruses are produced, and again, from the same apical membrane, the virus is gonna exit out into the lumen. So, right now, if you see here, the same apical membrane, the virus is gonna exit out into the lumen, and when you cough, or when you sneeze. So what is going to happen? This virus, when you cough or sneeze, again it is going to transmit to the next person through the respiratory droplets. So I hope you understood how the virus entered into your body and how it replicated in respiratory epithelial cells and how finally it went out in a bunch, right? So once this virus exits out, once this mini bunch of viruses, when they exit out in high number, and if one person is infected with this, now tell me what is going to happen, where this virus is going to land on him. So what I am going to do is that I am going to draw the same picture again and I am going to explain what is happening. For example, if I am infected with this virus got replicated in my respiratory epithelium, and let us say one virus entered inside, and right now I have I have synthesized around 100 viruses, I have replicated around 100 viruses, and right now I am coughing out. When I am coughing out or sneezing out, this virus goes and lands up onto the next person, and what is going to happen in that person I am going to draw it now. So let me draw the picture. Right. So if you see here, this is your nasopharynx, right? And this is your oropharynx. Nasopharynx and oropharynx together they join and form laryngopharynx. So this is your laryngopharynx over here, right? So I told you just for an example that we have got a tonsils over here. This is not the exact anatomical location, but for the sake of the picture, let us tell these are the tonsils over here. Now back of that you have got your blood vessel. So this is your blood vessel. This is your blood vessel, for example, right? I have explained you how the virus entered into your body, how the virus entered inside your respiratory epithelial cells, that is your columnar epithelial cells, and how the virus divided, right? And I have also explained you when the person will cough with such number of viruses, with such many viruses, and if these viruses land on your respiratory tract again on another individual, where it is gonna land directly at. So this virus can either land in your open cut mucosal surfaces or it can also land in your conjunctiva. So if they land in your open cut mucosal surface or conjunctiva, what is it going to do? This virus straight away will come down all the way. Let us say for example I have coughed and hundreds of viruses, they landed upon your mucosal surface, let us say oropharynx, right? Open cut surface of your oropharynx. So from that open cut surface, this virus, look here now very carefully, from that open cut surface, this virus landed on the oropharynx. Now from here, this virus will enter into my tonsils. Very important. This virus gonna enter into my tonsils. And when this virus will enter into my tonsils, what is it going to do? Let me draw the tonsils pretty big. So these are my tonsils over here, for example, right? So when this virus lands on my tonsils, so within these tonsils, my virus will very happily replicate like this. My virus is getting replicated in my tonsils. So what is the first thing that is happening, guys? What is the first thing? Once it enters into the conjunctiva, or let us say entry through the mucosal surfaces, what is it happening is that this virus can enter into my tonsils and replicate in my tonsils. This virus will replicate in my tonsils. It starts replicating in the tonsils. So once it starts replicating in the tonsils, what is it going to do? So from here, from the tonsillar area, this virus will jump into your bloodstream. From the tonsillar area, this virus is going to jump into your bloodstream. So right now the virus is within your bloodstream like this. Now it descends down all the way. Look here. Let me enlarge this blood vessel over here. right? It descends down all the way, and look here, you have got, for example, you have got your liver on your right side. On the right side of your body, you have got your liver, and on the left side of your body, let us say, you have got your spleen. This is your spleen, right? So this virus, look here, this virus goes all the way down like this, and it enters into the hepatic artery. After entering into the hepatic artery, this virus will straight away make its way to the liver, and this virus will enter into the splenic artery all the way like this, and finally enter into your spleen. So once it enters into the liver, what is it going to do? What is it going to do is that this virus starts replicating in the liver. This virus, it starts replicating in the liver. So there is heavy replication of this virus in the liver. Not only in the liver, even in the spleen also, this virus starts replicating. So till here, whatever I have discussed, what did I discuss? Tell me, what I have discussed here? Till here, what did I discuss? First, the virus entered into your respiratory tract, it replicated in the tonsils, right? So let me write down these things, guys. Then it entered into your blood, right? The virus then, it entered into your blood, and from the blood, this virus all the way came down and it went inside the hepatic artery and the splenic artery and entered into your liver. It entered into the organs like liver as well as spleen. And once after entering there, what did it do? There is a replication of this virus there also. Replication in the organs, right? Till here we have discussed. Now till here, whatever I have just told you, all these things, right? All the way from the replication in the tonsils till the replication in the organs, this entire process which I have told you, this is called as primary viremia. This is called primary viremia. All this thing over here is called as primary viremia. So now what is going to happen? What is going to happen is that, look here very carefully. Let me draw a few arteries over here, right? So again, this is the blood vascular system over here. This is a blood vascular system. So once the viruses started dividing in the liver, once the viruses started dividing in the spleen, don't you think it gonna affect the liver and it gonna also affect your spleen? It will definitely affect your liver and spleen. It will cause inflammation of your liver and inflammation of your spleen. So having caused this inflammation of your liver and spleen, this virus now starts entering into your normal blood vascular system. So now what is going to happen? Now this virus, once after dividing heavily, look now what it is going to do. This is the virus right now in your normal blood vascular system, and within your normal blood vascular system you have got your T cells, right? Now T cells, the moment it sees this virus, it will try to attack. But the interesting that is happening here is that this virus will enter inside the T cells and it will completely affect your T cells. Okay? Look here. For example, let us say this is your T cell. This is your T cell. This is another T cell. Let us say this is your third T cell. This is your fourth T cell. This is your fifth T cell. Now this virus can very happily enter inside the T cells and completely manipulate the T cells, take the charge over the T cells. Okay. So this virus right now is in your T cells. Okay. So once it is in my T cells, what is it going to do? It is going to completely damage my T cells. But before it damages my T cells, before it manipulates my T cells, what this virus is going to do is that this virus will order the T cells to go to the skin. Now this virus sitting within the T cells, it will order the T cells to go to the skin. So right now let me tell you that this is my blood vessel, for example, right? So this is my blood vessel that is supplying to my skin. And within my skin, you already know we have got different types of cells. For example, this is the basement membrane. You have got n number of cells. For example, these cells which I am drawing right now, right? Can anyone tell what is the name of these cells? These cells are called as keratinocytes. What are these cells called? Keratinocytes. There are different types of cells in your epidermis, but within comparatively to all the cells, 98% or 90% of the cells of your epidermis are made up of keratinocytes. Just 10% of the cells of your epidermis are made up of different kinds of cells. All these yellow color cells, all these green color cells which I have just drawn, these are called as keratinocytes. What are these cells called as? They are called keratinocytes. They are called keratinocytes. Okay. Now, within these keratinocytes, you have got some specialized cells. Look here. There are some specialized cells which are dark in color like this. You see, there are some cells which are really dark, and such dark colored cells are called as melanocytes. Such dark color cells are called as such dark color cells right over here, they are called as melanocytes. And not only that guys, not only that, 2 or 3 cells together, for example these 3 cells together, they form a special group of cells, and these are called as Langerhans cells. These are called as Langerhans cells. Langerhans cells. But whatever it is, out of melanocytes, Langerhans cells and all, there are some more cells, I am not mentioning it here. Out of all, 90% of the cells are keratinocytes. Keratinocytes are almost found everywhere in the epidermis. Okay, because in the epidermis you have studied from your histology that you have got stratum basale, stratum granulosum, stratum spinosum and all of that, right? So in all layers you have got these keratinocytes. Fine. So what is the big deal here? The big deal is that these T cells along with the virus enters into these keratinocytes. Look here. Look here now. This is your T cell over here. This is your T cell over here. Within this T cell you have got the virus. Within this T cell you have got the virus. Now what this virus will do is that this virus will move from one keratinocyte to another keratinocyte, infecting each keratinocyte. So this virus is going to move from one keratinocyte to the next keratinocyte. From here it will move to the third keratinocyte. From here it will move to the fourth keratinocyte. So in this way, this virus is going to move from one keratinocyte to the next keratinocyte. It means this virus will affect this keratinocyte, the second, the third and the first affected keratinocyte. So all these keratinocytes are affected in this way. All these keratinocytes are affected in this way. So when multiple keratinocytes are affected, what is it going to happen is that all these affected keratinocyte cells, they fuse together. Okay, let me write it here. All these are affected keratinocytes. Affected keratinocytes. So all these affected keratinocytes, they fuse together, and once after fusion, such type of cell you call it as, they form, all of them they fuse together to form a giant cell, and that the name of that giant cell is nothing but called as Zank cells. That is called as Zank cells. Clear? So they are called as Zank cells. So in this way, your entire epidermis, all over the body, your entire epidermis is getting affected. So the T cells, right, the viruses, they enter into the T cells and infect the T cells and take the T cells to the epidermis and cause damage of the epidermis. Okay, so in this way all the keratinocytes are affected. So the resultant of the affection of all these keratinocytes, why is it affecting all the keratinocytes? So whatever it is, it has got affected all the keratinocytes in this way. The resultant thing which you see on the skin is chickenpox. So this is the reason why the patient is going to land up having a condition called as chickenpox. So this is how the virus is going to cause chickenpox in the patient. So having discussed that how the patient has got chickenpox, so now you understood, I told you that this herpes virus, human herpes 3 virus will cause two diseases. One is called chickenpox, another one is called as herpes zoster, which is also called as shingles. So right now I have explained you how this is going to cause chickenpox, right? So till here, I mean from where guys? The replication has happened in the organs, and finally what has happened? The virus, they spilled into the blood again. So the replication has happened in the organs, then the virus spilled into the blood, back again, and from there what has happened, guys? From there what has happened is this virus entered into the T cells. What has happened? This virus entered into the T cells. I mean, it affected the T cells, and once after entering into the T cells, now this virus took its way all the way to the skin. So it finally went into the skin, and what has caused in the skin? Yes, what did it cause in the skin? It caused a condition called as chickenpox. So till here whatever I have discussed, all the way from the entry back into the blood till the affection of T cells, then taking it to the skin and causing chickenpox, this is called as secondary viremia. This is called as your secondary viremia. Okay. How much time will it take for primary viremia? For primary viremia, it would take around 4 to 6 days. 4 to 6 days it would take for primary viremia, and for the rash to happen, I mean the chickenpox to occur, for the secondary viremia part, it would take around 7 to 14 days. 7 to 14 days. Okay. The next important thing which you need to very well understand is that we have discussed how chickenpox has occurred. Now let us discuss how herpes zoster is going to occur. So how herpes zoster will occur in the patient? Herpes zoster, one very important thing you need to keep in mind is that, guys, let me explain you some basics, okay? Then I'll explain you how herpes zoster is going to occur. In the spinal cord, when you take the cut section of spinal cord, all of you know that this is how your spinal cord looks. This is how your spinal cord looks. So in this particular cut section of your spinal cord, if you very clearly examine here, few important things you can find. What are those few important things? Now you have got two different types of horns here. What are these two different types of horns, guys? Look here, these are called as your dorsal horns. On both the sides, these are called as your dorsal horns, and down below here, these are called as your ventral horns. Okay, that is called as your dorsal horn, this is called as your ventral horn. Now dorsal horns have got a ganglion, this is called as dorsal root ganglion. So what is happening overall? The dorsal horns all the way like this, the ventral horns all the way like this, the dorsal horns and the ventral horns fuse together to form a specific nerve, and this is what is called as your spinal nerve. This is what is called as your spinal nerve. Okay, and what is this particular guy called as? Dorsal root ganglion. Dorsal root ganglion. I hope you know what is dorsal root ganglion, right? So look here very carefully. Now in this picture, what I am going to mainly mention is that if you draw an imaginary line like this, the dorsal part is always sensory, the ventral part is always motor. The dorsal roots are sensory, it means it has got only and only sensory neurons. The ventral part is motor, it means it has got only and only motor neurons. So look here now, this is the sensory neuron like this. This is the sensory neuron. There are many thousands and thousands, but I am just going to draw few. This is a sensory neuron. Now from here I have got the motor neuron going out like this and entering into the spinal cord. So sensory neuron also, it is taking its way all the way into the spinal nerve. What did you understand by this picture? I understood that the dorsal horn along with the dorsal root ganglion, the dorsal roots are purely sensory. The ventral roots are purely motor. But the spinal nerve is a mixture of dorsal and sensory. That is the reason why from today onwards I call spinal nerve as mixed nerve. Why it should be called as mixed nerve? Because it has got both sensory neurons as well as motor neurons. Now if you understood this, now I am going to draw the real picture where I am going to explain you how herpes zoster is going to cause in this patient. Okay, so look at the next picture now. So this is just a rough picture, not an exact picture. Okay, so yeah, this is just a rough picture. Don't take it seriously. I mean, the margins and all of these might be wrong. Right. So this looks like a disabled person, but anyways, you know, concept is important. Right. So what I'm trying to explain you here, is that, guys, for example, let us say that here you have got your central nervous system. This is your brain. This is your central nervous system. So all the way here, this is your midbrain over here. Okay. After that, you have got your pons. You have got your medulla oblongata. Pons, medulla oblongata. And down all the way here, till here, all the way down, you have got your spinal cord. You have got your spinal cord. Okay. Now here exactly, few important things you need to know. What are these few important things? So here you have got a nucleus in the midbrain. This nucleus in the midbrain is called as mesencephalic nucleus. This is called as mesencephalic nucleus. The second important thing, you have got the next nucleus in the pons. This next nucleus in the pons is called as principal sensory nucleus. This is called principal sensory nucleus. And third important thing is that you have got a third nucleus right now attached to it in the medulla oblongata. So one nucleus you have got in the midbrain, second nucleus in the pons, third nucleus in the medulla oblongata, and this is called as a spinal nucleus. All these three together, they form a specific ganglion here. All these three, if you look, they form a specific ganglion here. Now this ganglion divides into three parts. One goes to the eyes, one goes to the maxilla, third one goes to the mandible. So this ganglion is supplying to three regions: ophthalmic region, maxillary region and mandibular region. So you call this ganglia as trigeminal ganglion. So this particular guy over here, this is called as your trigeminal ganglion. This is your trigeminal ganglion. And I have already told you that within the spinal cord you have got roots. I have already told you within the spinal cord you have got dorsal root like this, you have got ventral root. Dorsal root and ventral root. I'm going to draw a couple of them. So this is your dorsal root, ventral root, dorsal root, spinal nerve and ventral root, dorsal root, spinal nerve and ventral root. Okay. Now look here very carefully. For example, whenever let us say, and one important thing I forgot to mention here, what is that? Can anyone tell? In the dorsal root, I forgot to draw one very important thing. What is that? That is your dorsal root ganglion. So let me draw a bulge up. a bulge like this explaining dorsal root ganglion. So this is your dorsal root ganglion. Okay. So whenever your immune system, when this virus is rotating happily in your body and causing damage to your skin and causing chickenpox and all of these things, so that is a time when your immune system wakes up. Your immune system will become hyperactivated now. Lots and lots of immune mediators, lots and lots of cytokines, lots and lots of T cells, B cells and all of them, they come into action. So when there is hyperactivation of your immune system, that is the time this virus gets afraid and it is time for this virus to hide somewhere, right? So this virus retrogradely, I mean in the reverse way, this virus in a reverse way, look here, this virus in a reverse way, let us say, for example, let us say that this is your blood vessel over here. This is your blood vessel over here. Within that blood vessel, you have got your virus. This is your virus. Okay. Within your blood vessel, you got your virus. So with the virus within the blood vessel right now enters into your spinal cord. Okay. Through the spinal arteries and all, it enters into the spinal cord. So finally, what is happening? It enters into the spinal cord through the spinal arteries and all. So right now this virus, from here all the way, look at the arrows, it goes all the way and enters into your dorsal root ganglion. Directly it enters into your dorsal root ganglion. Not only the dorsal root ganglion, this virus even resides within your trigeminal ganglion here. Okay. So this is the virus residing in your trigeminal ganglion. Entering in your trigeminal ganglion. So this is the virus in your dorsal root ganglion and this is the virus in your trigeminal ganglion. It means, can I tell that this virus is going dormant? Can I tell that this virus goes dormant? Dormant in the sense hide for sleep. Where? In the spinal dorsal root ganglion plus it also goes dormant in where? The trigeminal ganglion. I mean plus the trigeminal ganglion as well. So it is hiding there in the dorsal root ganglion and the trigeminal ganglion. This virus hides. So once when the immune system again becomes weak, it will this virus waits a lot. It waits there, and finally one or the other day the patient will be affected with some sort of disease, maybe cold, maybe cough, or let us say maybe he will have some kind of bacterial infection or whatever it is. So in that case, when the person becomes weak, when the immune system of the person is too weak, right, that is the time this virus will know that, and suddenly from exactly how it went inside the dorsal root ganglion, the way it goes inside the dorsal root ganglion, this is called as a retrograde way. Now from the dorsal root ganglion it will come back and this is called as antegrade way. So from in an antegrade fashion, from the trigeminal ganglion and from the dorsal root ganglion, the virus is going to come back. Look here, the virus is going to come back and settle here near the skin. The virus is going to come back and settle near the skin. The virus is going to come back and settle near the skin. So it will settle near the skin all the way like this till here. I mean from here it starts its rash. It will settle on the skin and it will initiate a rash on the skin, and this rash starts from here, it goes all the way here till the center, or starts here, it goes all the way till here. But remember one thing, that this rash will not cross to the next dermatome. It will not cross to the opposite side. If the rash is on the same side, if the virus is on the same side, the rash also will be only on the same side. It will be along, we call this as the infection is along a single dermatome. This infection will not cross to the opposite side. It will be on the same side. If you look at the picture of the patient suffering with this virus, you will see the infection or the vesicles or the skin rash, right, in the form of vesicles, they are formed only on one side of the body, not on the other side, either right or other left, along a single dermatome. If the infection is on the head, right, through this trigeminal ganglion, it will be only on one side, not on two sides. It means it is only along the single dermatome. If it is on the ear, it will be only on one side of the ear, single dermatome. Okay, so that is a characteristic appearance of this rash. If someone asks you, what is this condition basically? Okay, when your immune system becomes weak, when your immune system becomes weak, then this entire thing gonna follow antegrade and settle on the skin and cause a specific infection that is called as herpes zoster or shingles. Herpes zoster or also called as shingles. So if someone asks you, how can you define shingles? What is the characteristic appearance of shingles? The characteristic appearance of shingles is that shingles, keep in mind one thing, it will stay along a single dermatome. Shingles stay along single dermatome. It does not cross to the opposite dermatome. Okay, so this entire process where shingles is formed, this is called as a reactivation of the virus. Primarily the virus was sleeping, now it got reactivated and came all the way back and caused this type of pathology. Okay, now another important thing which you need to know, two important things I have already told you, that this virus is going to cause shingles or chickenpox, and other way it is going to cause herpes zoster, right? Third important thing which you need to know is that this virus, I told you in the starting, during the primary viremia state, this virus affects your liver. It causes inflammation of your liver, and when it causes inflammation of your liver, that condition, my friend, you call it as what? Hepatitis. So this virus basically causes what? Hepatitis in the patient. Okay, not only hepatitis, look here, if I draw it little big, I mean this is your laryngopharynx, trachea, these are for example your lungs over here, these are your lungs over here. This virus can very easily spill into the lungs, it can very easily enter into your respiratory tract and cause infection within your lungs, and this infection you call it as pneumonia. You call it as pneumonia. What is this infection? You call it as pneumonia. Not only that, this virus can even enter into your central nervous system and cause damage to the meninges, damage to the brain. So damage to the meninges and damage to the brain, together you call it as encephalomeningitis. We call this condition as encephalomeningitis. Pneumonia, encephalomeningitis and hepatitis. It causes basically these three things, apart from the chickenpox one and the herpes zoster, that is the shingles. Okay, so always and always again remember that if I ask you about the shingles, you have to tell that shingles stay only, the characteristic rash in shingles is it stays only along the single dermatome. But anyways guys, just for the note's sake, we have to definitely write it. How does a chickenpox rash look? How does a chickenpox rash look? The basic way how does it look is that this is a vesicular rash. This is a vesicular rash. So what do you mean by vesicular rash? So that rash which is very itchy, okay, that rash which is very itchy, the patient will have very much of itchiness, right? Not only itchiness, if you ask me in chickenpox, how does the rash spread? It starts from the head, trunk and then extremities. It means it is centrifugal, going away, right? Such type of rash is centrifugal rash. What type of rash is centrifugal rash? Such type of rash which goes from, it starts from the head and neck, I mean the trunk, and goes to the extremities. So, head, trunk to extremities. Head, trunk to extremities is this type of vesicular rash. I mean the chickenpox rash, right? So, next thing is that herpes zoster. Coming to the second form of rash, that is your herpes zoster. So, in herpes zoster, what is this type of rash? How do you define this rash? I have already told you this rash is only along the single dermatome, right? Only along the single dermatome. So, you call it as unilateral rash. Unilateral vesicular rash, not only vesicular, it is also pustular. Pustular and only along the single dermatome. Only along single dermatome, only along the single dermatome. And such type of rash, it do not cross the midline. It do not cross the midline. Right? Do not cross the midline. Third important thing is that in many unfortunate cases, this rash can even affect your eyes. This rash can even affect your eyes, right? So, when it affects your eyes, you call this condition as herpes zoster ophthalmicus. This condition is called herpes zoster ophthalmicus. Now, if someone is going to ask you what is this herpes zoster ophthalmicus or which nerve is affected in this herpes zoster ophthalmicus, you have to tell that the nerve which is affected in this herpes zoster ophthalmicus is fifth pair of cranial nerve, right? Nerve which is affected here is fifth pair of cranial nerve. In the same way, in some individuals, you will also see the rash that is affecting the ear of this patient. Okay, so this rash gonna even affect the ear of the patient. So this is called as herpes zoster oticus. Herpes zoster oticus. Okay, so this condition is called as herpes zoster oticus, where which pair of cranial nerve is going to be affected, guys, in herpes zoster oticus? That is eighth pair of cranial nerve, that is your vestibulocochlear nerve. Eighth pair of cranial nerve, that is your vestibulocochlear nerve, and this condition is also called as Ramsay-Hunt syndrome. Ramsay-Hunt syndrome. Ramsay-Hunt syndrome. So, how do I diagnose this case? How do I diagnose such patient of herpes zoster? How do I do a diagnosis is that I told you I will see some Zank cells, right? So, where will I see them? On Zank smear. On Zank smear. Second important thing is by PCR. All of you know PCR is most commonly used. So, this is the end of the lecture, guys. Thank you.